very pleased to be here today, and indeed I'm going to take you to a slightly different kind of story. As we all know, the expansion of the armamentarium to control Helminth infection is urgently needed. And so is the acquisition of a better, deeper understanding of the mode of actions of the currently used anthelmintics. So with this, I will take you through the very recently published opinion paper that was called exactly as this presentation, When Secretomes Meet Anthelmintics, Lessons for Therapeutic Interventions. So to survive in their hosts, Helminths have evolved a range of strategies to remain there for months or even years. And you can imagine the host environment is not a very hospitable and friendly one when there is an engaged immune response against the pathogen. So there has been a molecular negotiations between host and parasites. And this has resulted in highly specialized interspecific relationships in which the secretomes may delimit conditions that render a host permissive for an infection to take place. So what are these secretomes? These are basically any kinds of molecules that are released either by excretion or secretion by a parasite into the host environment. And these may be proteins, nucleic acids, lipids, small metabolites, glycans, and basically anything. And this also includes extracellular vesicles, which is a very fancy research topic in molecular helminthology nowadays. These are small membranous structures that are released by any kind of cell. Virtually any eukaryotic cell releases extracellular vesicles, and these are like small compartments encapsulating all these other kinds of proteins, nucleic acids, and other molecules. And so many of these secreted molecules or excreted molecules have the capacity to modulate host immune responses. And these have attracted, obviously, increasing attention. So these secreted proteins have consequences, functional consequences for host cell functions. And these consequences are very varied. It really depends on the type of cell we talk about and also the type of pathogen we talk about. But obviously, secretomes are a promising source of drug targets, both for drugs and vaccine discovery. So obviously, identifying parasite-derived molecules that render a host permissive for an infection to take place should encourage efforts to rationally target these important key components to restore the host's capacity to either resist or expel, eliminate the infecting worm. So an anthelmintic drug discovery in real life is done like this. So initially, it was done in the early years, in the 70s and so on, by simple, if I may say so, empiric observation of efficacies in vivo, so in an infected animal. And this has shifted slightly now to more modern days to phenotypic screens, mostly ex vivo, in vitro, where the viability of a worm is observed as a consequence of exposure to a drug or a compound. This has been complemented partly, at least, by mechanism-based screens. So this involves the availability of recombinant systems and powerful in vitro tools. This is, however, slightly limited by the fact that the mode of action of many drugs and compounds are unknown. But screening a little bit the literature, we noted that there was an underappreciated characteristic of some anthelmintics, that is their ability to block the release of these parasite-derived molecules, so that these anthelmintics are basically capable of disrupting this dialogue from the parasite to the host. Do anthelmintics act by disrupting parasite secretory or excretory processes? Well, basically, the characterization of parasite-derived molecules has led to the concept that therapies can be devised to disrupt the dialogue between parasite and host, and work on vaccines that specifically target secreted antigens based on their known or just presumed roles in enabling an infection has shown that this approach is valid and can generate therapeutically useful outcomes. Now, there is evidence out there in the literature regarding the macrocyclic lactones, so I'm going to talk about ivermectin again. And this class of drugs acts by interfering with Helminth nervous system by preventing responses to excitatory neurotransmission. And this generally results in an impaired muscle function and paralysis of the worm. Where the benzimidazoles, where albendazole and mebendazole are classified, act rather by inhibiting the microtubule polymerization. And this results in deep physiological alterations of really multiple, basically any processes, and that includes secretion. So the absence of stable microtubules hinders the intracellular trafficking of these parasite-derived molecules and their ultimate secretion. If the mode of action of some anthelmintics seems to impair aspects of parasitic nematodes that control these molecule releases, then the effects on secretion seem to be correlated very well with the spectrum of activity and the kinetics of expulsion of infecting worms, as well as with the levels of drug exposure that is achieved in vivo. At least this was shown for a few nematode species and a few stages. But whether inhibiting secretion is the key trigger for parasite clearance or one of several pharmacological effects remains to be determined. The benzimidazoles have a only not very obvious effect on adult worms of several nematode species. So where the parasite motility, their viability remains largely unaffected. In fact, it is a disruption of parasite secretory processes that seems to be the earliest physiological effect of this drug class exposure. This is translated or illustrated by the accumulation of secretory granules along with disappearance of microtubular structures. That's what observed at least in Ascarisum. And there is also an accumulation of certain proteins, such as acetylcholinesterase in parasites. And this was noted along with a marked suppression of the release in vitro of this very protein. So these effects observed with benzimidazoles at least applies very well to gastrointestinal nematodes. The situation is a little bit different for tissue-dwelling nematodes because the expulsion kinetics are partly the result of the duration of exposure to effective drug concentrations. Plus, the action of benzimidazoles is reversible. So the pattern of exposure to benzimidazoles and the dynamics of parasite expulsion are compatible with the contribution of disrupted secretory processes as a cause of parasite elimination. Now, for the macrocyclic lactones, these mainly kill or affect developing stages, so microfilaria in filarial nematodes or embryos. And there is evidence that has accumulated to support that the role of the ivermectin targets, the glutamate-gated chloride channels, in parasite-derived molecule release via the control of neuromuscular activity of the secretory system. And it was shown that in Burgdorferi microfilaria, these ion channels are associated with a structure surrounding the secretory vesicles. And this is proposed to control molecule release. Now, both protein and extracellular vesicles release is inhibited by macrocyclic lactones. There is now even more evidence accumulating regarding the effect on these class of drugs and the production and release and the contents of these extracellular vesicles. Now, what we suggested is that the identification of key parasite-derived molecules as determinant of the equilibrium between host and parasite, as well as the mechanisms that control their release, will likely expand our list of targets that can be exploited for therapeutic interventions. Now, the evidence from pharmacology and immunoparasitology as well should be reconsidered and put together to inspire the search for novel ways to interfere in the communication between parasite and host. And we would like to recommend that efforts be made to incorporate screens for inhibition of these parasite-derived molecule release in culture as a complement to existing phenotypic screens. And there are very famous technical challenges to do this that may limit our ability to pursue this goal. And this is, for example, our incapacity to maintain the complete life cycles of these nematodes in vitro. We still heavily rely on animal models. And our current in vitro systems are flawed, but these are the best we have. And we'll also recommend that expanding the scope of discovery programs so that these integration of product release and phenotypic screens may help really greatly expanding the scope of discovery program to be able to detect such compounds. And that is going to be an investment, but that should be an efficient one to bring a new compound into the pipeline. I'd like to finish with a couple of outstanding questions, of course, that should or hopefully will shape research priorities regarding new anthelmintic discovery. Can we ultimately reduce Helminth secretory systems to an in vitro model to complement currently used phenotypic screens, so in vitro systems? Will it be possible at all to reconstitute the host-parasite crosstalk using in vitro systems and maybe applying a stress situation that mimics what the host does to the parasite to reach a setting as natural as possible, and this to be integrated in anthelmintic screens? And can we develop tools for functional genomics of nematodes that will facilitate the characterization of parasite-derived molecule secretion biology as well, and then identify, prioritize molecules that are essential for establishing and maintaining an infection? Now you can find basically all I said in the paper entitled When Secretomes Meet Anthelmintics, Lessons for Therapeutic Interventions, which is published in Trends in Parasitology. And I would like to thank very much my co-authors, Dr. Giovanni Moreno and Professor Tim Geary, as well as our funders, and I'm very grateful for your attention.